And so
Thank you. Thank you for reminding me.
you're welcome.
All right, so today's lesson is going to be, we're going to talk about eukaryotes, specifically just plants and animals. And then we're going to talk about osmosis and plasma membranes. So last time when we talked about the difference between prokaryotes and eukaryotes, just in general, and this is from our notes from last time, we said that prokaryotes are all bacteria and that they are smaller, simple cells, whereas eukaryotes can be plants, animals, fungi, or protists, but they are larger, more complex, and then their DNA is different. Their DNA is different. That their DNA has these things called histones associated with it, and their DNA is also located inside of a bag within the cell. So just like last time, we drew a picture of a typical prokaryote. Last time we were here, we drew this gorgeous picture of a typical bacterial cell. Today, we're going to draw a plant and an animal cell and label their parts. There is a diagram in your book, and I'm looking for a page number, and I don't have my book open, but I told you guys last time, oh, here it is. Pay attention in your book, figures, and I don't have a page number. If anybody has a page number, let me know, please. These two diagrams in your book, figure 4.6 and 4.7, make sure you know their names of these structures. That we're going to label. So I'm not going to write out what their function is, but you can look it up on the diagram. So we'll start out drawing our plant cell. I'm going to need some different colored markers today. All right. So starting first with my plant cell, and I'm going to try and draw them both on this same sheet of paper here. My plant cell, I'm going to draw it kind of squarish. And then I'm going to give it a little thingy like this. Okay. So here I am drawing my plant cell. The first structure I'm going to label is that square on the inside. That is my plasma membrane. And then just outside of the plasma membrane is the cell wall. So one big difference between plant and animal cells right off the bat is that plant cells have a cell wall. So I'm going to draw an animal cell here at the bottom. And I'm going to draw him kind of roundish. And this is right here, this is my plasma membrane on my plant cell. But first thing is that the plant cell has a cell wall, whereas the animal cell does not. And I guess I could label this animal cell. And I'll label this as a plant cell. So the cell wall, this is not exactly the same as a cell wall as a bacterial cell, but it has a similar function in that it's for protection. The plasma membrane, you can think of it as like the skin of the cell, so to speak. This is what holds everything inside of the cell. Same thing for the animal cell. It's kind of like its skin. It's what separates the outside world from the inside of the cell, the plasma membrane. Now, filling up both cells, they both have this watery gel stuff, which I'm just going to draw as some lines here. They both contain cytoplasm, which is that watery gel that fills up the interior of cells. So they both contain cytoplasm. The most prominent thing that you can see inside of both plant and animal cells is this big, huge structure called the nucleus. So I'm going to draw a nucleus here. Both plant and animal cells contain what's called a nucleus. The nucleus, this is the bag which holds the DNA of the cell. So this is literally a bag, and it's actually a double bag. There's two bags. It's a bag inside of a bag. And the bags are not perfectly solid. They have all these tiny little holes. But that's where you're going to find the genetic material or the DNA of your plant and your animal cell is inside the nucleus. Continuous with the nucleus, so in other words, it touches the nucleus and it comes kind of off of it like this. You have what looks like a ribbon looking material. Both the plant and the animal cell have it. That ribbon looking material is referred to, and I'm just going to abbreviate it as the ER. I have more room over here, so I'll spell it out. The Endoplasmic Reticulum. And so the ER, what it is, it's a series of tunnels which allows things to travel through the cell. There's two types of ER. There's the rough, and then there's the smooth. You can tell the difference between them. The rough is closer to the nucleus, and it has all these tiny little embedded ribosomes in the surface. And so it looks bumpy or rough. The smooth ER doesn't have any of those ribosomes, so kind of the opposite of rough would be smooth. Even though they're both tunnel systems, the presence of the ribosomes kind of makes them do something a little bit different. The rough is helpful in making proteins. The smooth makes fats. And it's also important in detoxification or removing poisons from the cell. You can have ribosomes floating freely. They don't have to be attached to the ER. They can be floating freely also. So both plants and animals will have what we call free ribosomes. They'll also both have somewhere inside of them something that looks kind of like a stack of pancakes. That little stack of pancake thing is called the Golgi body. anytime a cell is going to be secreting something, it's going to have the help of the Golgi body to help secrete whatever that is. So if you can imagine a cell that has to produce wax or a cell that has to produce mucus, if the cell is going to produce something that it kicks out of the cell, it's going to be the job of the Golgi body to help it get out of the cell. One of my favorites, if you can have a favorite, one of my favorite cell structures is what's called the mitochondrion. Both cells have mitochondria in them. Mitochondria are always drawn like this, kind of a kidney bean shape, the wiggly line on the inside. But this is my absolute favorite organelle. That's the mitochondria. His job is to make energy. And remember that cellular energy is this stuff called ATP. So the whole job of the mitochondria is to make ATP. It breaks down sugar in order to do that. So both cells have mitochondria. So far in our diagrams, our cells are exactly the same, except for the presence of the cell wall. So you might want to put a star by the cell wall to remind yourself that that's in plants only. But every other structure that we've drawn, you have both in plants and animals. I'm going to draw two more structures that you find in plants only. Two more structures that you find in plants only. One of them is kind of like the mitochondria, kind of like it. It's this beautifully green structure, which has another bag inside of it, and that is called the chloroplast. This is what makes plants green. And the chloroplast, its job is to make energy, but by way of photosynthesis. Whereas the mitochondria makes ATP by way of aerobic respiration, a different set of chemical reactions. So they both make energy, they just do it differently. They just do it differently. Of course, animal cells don't have chloroplasts. The other structure that we see in plants only is a very large bag of water. A very large bag of water called the central vacuole. Again, that's in plants only. It's this huge bag of water inside of plant cells. It helps plants to stand up by way of what's called turgor pressure. By way of turgor pressure. Turgor pressure is internal water pressure. So plants don't have bones like animals do, but they do have skeletons in a sense, and their skeletons is made up of water pressure. So they have a water pressure skeleton, and that skeleton, so to speak, is made up of all these tiny little central vacuoles, or these bags inside of plant cells. The last structure I'm going to mention is found in animals only. So animals have a special structure that we only see during cell division. And these tiny little structures, which I'm just going to draw them almost like a little letter L, these tiny structures that we only see during cell division, these are called centrioles. And we only see them in animals. Plants don't have them. So one of my favorite quiz questions is to ask what structures are found only in plants, what structures are only found in animals. Make sure that you review that information. What's found only in plants and what's found only in animals, because I promise you that's going to be a quiz question. So all these structures that we've been labeling are referred to as little organs or as organelles. All these structures that we've been labeling little organs or organelles. Before I move on, let me check and see if you have any questions for me.
No, I don't have any questions.
Thanks, Rachel.
You're welcome.
All right. So we'll go on to the next topic, which is what is osmosis? Well, osmosis is a special type of diffusion. And diffusion, in case you're wondering what that is when molecules naturally move down a concentration gradient. So diffusion is when molecules move down a concentration gradient. And all that means is, let's say over here you have a bunch of perfume molecules. Over time, they spread out. And so moving down a concentration gradient means to go from an area of high concentration, where they're packed together real tightly, to an area of low concentration. Or it means things have a tendency to spread out. Things have a tendency to spread out. So diffusion is just molecules, they have a tendency to spread out, or they go from an area of high to low concentration. Or you can say they move down a gradient. Well, osmosis involves spreading out. But because it's a special type of diffusion, what are we exactly talking about? Osmosis specifically is the diffusion of water but through what's called a selectively permeable membrane. So diffusion is just a very simple explanation about spreading out. Molecules, they spread out. But osmosis is specifically the spreading out of water, and it involves something called a selectively permeable membrane. And this is an important concept in biology because a lot of things enter and exit the cell either by diffusion or by osmosis. So the way things that have to get in and out of the cell is through the plasma membrane. So if we look at the plasma membrane of the cell more closely, we'll find that it's an example of something that's selectively permeable. That means some things can pass, but others cannot. So some substances can pass through, but others cannot. That's what we mean by selectively permeable. Some things can pass, other things cannot. So we'll draw a little diagram, a close-up view of the plasma membrane. The plasma membrane itself is made mostly of phospholipids. Okay, it's made mostly of phospholipids, but it also contains some sugars, and it also contains some proteins. But by far, the biggest component is this special type of fat called phospholipids. So a phospholipid all by itself looks like this. We usually draw a circle with two tails. And phospholipids are very interesting in that they're the only type of fat that partially dissolves in water. So the head of this fat right here is what we call non-polar, but the tails are what we call polar. And if something is polar, it dissolves in water. So the heads won't dissolve in water, but the tails do. So phospholipids are very unique fats. They're the only ones that will partially dissolve in water. So if you take a bunch of phospholipids and you line them up together, an interesting thing happens. Is they naturally orient themselves so that the heads dissolve into each other. And they naturally orient themselves so that the tails dissolve into each other. And then Mother Nature took a second layer of phospholipids and she put it back to back with the first layer. And again, the heads are oriented so that they dissolve into each other. And the tails are oriented so that they dissolve into each other. Oh, you guys, I made a mistake. I made a mistake. Okay, hold on. Let me correct myself. I made a mistake. Right here. Let's fix your notes right here. I said this backwards. I said this backwards. The heads are polar and the tails are non-polar. I'm so sorry I said those backwards. The heads are polar and the tails are non-polar. I think I need more coffee. I'm still thinking I should be in bed under the covers. Okay, so let's label this correctly. The heads are polar, and that means that they dissolve in water. But the tails are non-polar. So if something's polar, it dissolves in water. If it's non-polar, it won't. There we go. Okay, glad we fixed that. How embarrassing. Okay, this arrangement of two layers of phospholipids, that is like the basis for building a plasma membrane. This is what's called a phospholipid bilayer. That's what's called a phospholipid bilayer. Plasma membranes are made up of a phospholipid bilayer. The way things can get in and out of the cell, part of it has to do with the chemistry of this polar versus nonpolar business, and part of it has to do with the space or the tiny little pores between the phospholipids. So some things can pass while other things are blocked from going in. Now, I mentioned there are other things as part of the phospholipid layer. There's some proteins. There's proteins and there's carbohydrates. We'll draw those in. Some of the proteins are very large and they go all the way across like this. Okay. Some of the proteins are relatively small and they stay on the top. And then the carbohydrates are usually attached like this to a protein. So we'll see proteins that are embedded in the phospholipid, and we'll see like little carbohydrates that are attached. And it makes a really elegant picture, a really elegant diagram. The plasma membrane of the cell is often described as the fluid mosaic model. You often hear it described as a fluid mosaic. So where do they get this name, this fluid mosaic? Well, the reason they call it a fluid mosaic they get the word fluid because the phospholipids are not stiff. They are flexible and they have fluid-like movement. So scientists, they don't imagine this to be a very stiff, solid structure. Instead, they imagine these tails are constantly kind of moving around. I don't know if you can see my fingers, but they're constantly, like moving around. They have some flexibility to them. They're kind of in motion all the time. And then they get the word mosaic because there are so many parts to this big picture, which is a big picture made of smaller pictures. So in other words, you can make up a picture of just a phospholipid. You can make up a picture of just a protein. You can make up a picture of just a carbohydrate. But then you take those pieces and put them together and make up a larger picture. So you can have a single pic of just a phospholipid by itself. You can have a single pic of one protein, and you can have a single picture of a carbohydrate, but you combine all these single pictures into one big picture. And so that's where we get the word mosaic from. It's a big picture made up of a lot of smaller pictures. So when someone asks you, what does a cell membrane look like? Well, we often say, well, it's a fluid mosaic because the phospholipids are flexible, but it's also a big picture made up of a lot of smaller pictures. You'll probably see a question on the quiz about what does it mean to have a fluid mosaic. If you would type a number 1 for me into the chat window when I can move to the next idea. Good, good. Y'all are keeping up. Good. So this description of the plasma membrane that we've just done, both plant, animal, and bacteria cell membranes are built like this. So I'm going to make a note of that here. Prokaryotes and eukaryotes have similar plasma membranes. They're not exactly the same, but they are similar and that they are both described as fluid mosaics. Okay, they're both described as fluid mosaics, meaning they're made up of phospholipids, various sugars, and various proteins. They differ in the types of sugars and the types of proteins, but they are similar in that they have, they're both made up of fluid mosaics. All right, so now back to this idea of osmosis. Back to this idea of osmosis. Osmosis, what it requires for it to occur, what it requires is you have to have a selectively permeable membrane. You also have to have an impermeable substance. So something that cannot cross. And you have to have, of course, a concentration gradient. And you got to have some water. So I'll show you what I mean. Let's say right here, that's my selectively permeable membrane. That's my selectively permeable membrane, and it's separating two solutions. It's separating two solutions. On this side of the membrane, I have, let's say, 20% sugar. And so if I have 20% sugar, that leaves me with 80% water. And on this side of the membrane, I have 10% sugar. And that leaves me with 90% water. So looking at my requirements, I've got a selectively permeable membrane. Do I have an impermeable substance? Well, in this example, Water can cross freely, but the sugar cannot. So the sugar is going to be the example of something that is impermeable, meaning that the sugar cannot cross this membrane. This membrane has all kinds of little holes in it, but the holes are too small to allow the sugar to cross. So the sugar cannot cross. In this example, my sugar is going to be impermeable. Do I have a concentration gradient? In other words, is there a difference in the amount of stuff on both sides of the membrane? Tell me out loud, do I have a concentration gradient on this side and that side versus that side? Tell me yes or no out loud. Sometimes I feel lonely, you guys. I need to hear your voice.
No.
I'm so glad I
No.
heard your voice, Brandon. But try again. Do I have a concentration gradient? In other words, is there a difference?
No.
You guys are hilarious. Yes, I have a concentration gradient.
That's what I meant. I meant yes.
I know you meant yes. Yes, I have a concentration gradient. That means that there's a difference in the amount of stuff I have on both sides. I have a sugar gradient. I have more sugar over here than what I do on this side. And then I have a water gradient. I have more water on this side than what I do on this side. So gradient just means there's a difference. So I actually have two gradients, a sugar one and a water one. Now, things like to be equal on both sides of a membrane. In other words, the law of diffusion says things like to spread out. But because I have a membrane here, the sugar cannot spread out. In other words, the sugar can't cross. The sugar can't cross. It can't cross this way and it can't cross that way. But the water can cross no problem. It can go back and forth. What ends up happening is that the water is going to try and dilute the sugar. In other words, water always goes from high to low. That's what relieves this, in other words, this osmotic pressure. Because this side has a higher sugar, it has what we call higher osmotic pressure. And water always goes from high concentration to low concentration, or water always goes towards the higher osmotic pressure. Water always moves towards higher osmotic pressure. So what that means in terms of osmosis is that some water is going to come in this direction, but so much more water is going to be going in the opposite direction. So we say the net movement of water is going to be towards this side. So eventually this side is going to lose water and this side is going to gain water. I'll give you another example. Same kind of situation. Same kind of situation. Here's our selectively permeable membrane. And we're going to separate it this time with a 15% sugar solution. And that will give us 85% water. This side's going to have a 25% sugar solution, 75% water. In this example, we'll call this solution A and we'll call this solution B. And so tell me, which solution has higher osmotic pressure?
Would it be solution B?
It is
Or-
solution B. Why did you answer B?
Because in solution A, the sugar is 15%, and in solution B, the sugar is 25%.
So
And
the
then
okay, keep going.
because 85% in solution A of water wouldn't that dilute the sugar a little bit? And
Yes,
so,
but I just want to know why did this one has higher osmotic pressure? Because B, and so this is like if this was a quiz question, this would be the answer that you would look for as a multiple choice answer. B has a higher osmotic pressure because it has more what? What gives something a higher osmotic pressure?
would it be the sugar? Like
Yeah.
the amount of sugar?
There you go. B has more sugar. There we go. And sugar is the source of the osmotic pressure. So more sugar means higher osmotic pressure. So very good. So here's question number 2. Is there a concentration gradient present? Is there a concentration gradient present? Yes or no? Brandon, this is your question. Is there a concentration gradient present between these two?
I got it wrong this time, but I'm gonna go ahead and say yes.
Yes, there is. There's actually 2 of them. What are the 2 gradients?
Okay.
One is for the sugar.
And one water.
Uh-huh. Yes, there is a sugar gradient. And then there's a water gradient. Remember, the word gradient just means there's a difference in concentrations or how much is present. So because there's different amounts of water and there's different amounts of sugar, yes, there's actually 2 gradients.
And at the bottom it says difference in what?
Amount present. Oh, sorry, amount present. So here's another question. Which solution has the higher sugar concentration, A or B? Which one's got more sugar, A or B?
B.
Yeah, B. B. So because B has more sugar. That's another way of saying which one has the higher osmotic pressure. We're going to use a word to describe B compared to A. We're going to say that B is hypertonic to A. The word tonic refers to osmotic pressure. And hyper means elevated. So B has elevated sugar, or it has greater osmotic pressure. That makes letter A hypotonic as compared to B. Because it has less osmotic pressure, less solids. So my last question, what is the net direction of osmosis? Is it going to be from solution A into B, or is it going to be from B into A? In other words, which way is most of the water going to move? What's the net direction of osmosis? We know that the sugar cannot cross, so the sugar cannot cross. But we know that the water can go back and forth freely. Which way is the water going to flow? Is it going to flow mostly into A, or is it going to flow mostly into B? Tell me out loud.
Gonna flow mostly into B.
Yeah, very good. It always goes towards higher osmotic pressure. So the last thing that I'll share with you, these words hypertonic, hypotonic, isotonic. Let me know when I can go on to the next page. Type a number 1 for me into the chat window. Good, good. So 3 terms, hypertonic, hypotonic, and isotonic. When you're using words to compare, let's say, let me do it this way. No, that's the word. Say here's a solution A, here's a solution B. If this is 10% sugar, and this is 20% sugar, then we say that B is considered hypertonic and A is considered hypotonic. Because the words hyper and hypotonic, they represent the stuff that's dissolved in the water. So hypotonic always has fewer solutes or fewer things dissolved. In this case, sugar. Hypertonic would be you have more solutes or more sugars. Okay, so hypertonic solutions always have a higher osmotic pressure. These always have a lower osmotic pressure. If you have 2 solutions, let's say this is solution C and D, and they're both the same, 40% sugar, 40% sugar. What happens if they're the same? If they have the same amount of solutes, then they are isotonic to each other. Uh-oh. They are isotonic to each other. If they were separated by a membrane, there's no net movement of osmosis. In this situation, it always flows from, it always goes to higher osmotic pressure, the net direction. All right, I'm about out of time, but I wanted to make sure that I use these words for you because you'll see them on the quiz. Any questions for me as we're wrapping up today's session?
No, ma'am.
No questions.
Oh, you guys are so smart. So I'm going to turn the recording off